Welcome student friends in Alka's Botany channel. I am Dr. Alka Inamdr in Alka's Botany channel. Please like, share and subscribe the channel. Mitosis has a history in which Turnip in 1826 reported cell division, and then in 1835 von Mohl described the cell division. This means the process of cell divisions was identified and described even before the discovery of laws of inheritance by Mendel. In 1846, K. Nageli discovered the new plant cells arises from pre-existing cells, and later on in 1855, R. Virchow discovered that all cells arises from pre-existing cells. We have studied all that in cell theory. Confirmed that the cells divide and produce new cells. In the year 1880, W. Fleming introduced the term mitosis, and in 1882, Strasburger, a German botanist and now called as a father of modern botany, described the mitosis in plant cells. So cell division is a complex phenomenon by which cellular material is divided equally between daughter cells. The cell division in eukaryotic organism consists of two integral processes, nuclear division and cytoplasmic division. Nuclear division is called karyokinesis and cytoplasmic division is called cytokinesis. The cell that undergoes division is termed as parent cell, while the cells formed by the division in parent cell are called daughter cells. The chromosomes are duplicated and distributed equally to the daughter cells is called mitosis. It is also called somatic cell division. So mitosis is the process of cell division whereby the chromosomes are duplicated and distributed equally to the daughter cells. The mitosis is a progressive, continuous process, but on the basis of significant changes and for the sake of convenience, it is divided into four main phases. Those stages are prophase, means early, metaphase, that is called middle figure, Anaphase is called up and down figure, and telophase, it is the final figure. Like that, there are these four phases, which is nothing but a division phase of cell cycle after completion of interphase. In prophase, the cell begins the process of division in which many structural changes takes place. The chromosomes condense, which makes them easier to pull apart. The nuclear envelope disappears. The double membrane that surround the nucleus dissolves into a collection of small vesicles. Nucleolus disappears by the onset of prophase. The centrosomes move to the opposite poles. During interphase, the pair of centrosomes were together just outside the nucleus. In prophase, they separate and move to the opposite ends of the cell. The spindle starts to form, growing out of the centrosomes towards the chromosomes. This prophase is the longest phase of mitosis, where there is chromatin material which is inside the nucleus. It is unwound form of the chromosome. It starts condensation, and due to that condensed material, the tightly wound, or it is called as a coiled form of the chromosome. Like that, there are coiling of chromosome takes place during the process of prophase. Thus, chromosomes that have duplicated will have two sister chromatids attached to the same centromere. These are called as sister chromatids which have identical genetic information. This is a prophase in which chromosomes condense, that is no longer chromatin, but these are in the condensed form, which are easily understood that there are two sister chromatids attached with each other by the centromere, and these are visible in prophase in the late prophase. Nuclear envelope starts disappear in the late prophase. Centrioles move to the poles of the cell and spindle fibers begin to extend from the poles. In this image, we can easily understood that in early prophase, there is a coiling or chromatin material is as it is and nuclear membrane is also there. But in the late prophase, this nuclear membrane and nucleolus disappears and coiling of chromosomes are very well identified. That means nuclear envelope disappearing, centrioles moving to the poles and spindle fibers are formed. These spindle fibers are nothing but a microtubules about 250 angstrom unit in diameter, and microtubules are cylinders formed from the protein monomers. These microtubules varies from 16 to 5000 in this higher plant. Towards the end of prophase, the nuclear membrane disintegrates and its contents are dispersed in the cytoplasm. The chromosome starts moving toward the equatorial line and there is an end of prophase takes place and it starts the metaphase. Metaphase lasts only for a short time of nuclear division cycle. The chromosomes reach the central or equatorial region of the spindle. They are lined up in one plane to form the nucleoplasmic plate or equatorial plate, which is right angle to the plane of a spindle. The smaller chromosomes usually remains towards the interior of the spindle while larger chromosomes remains at the peripheral position. The arms of the chromosomes remains free in the cytoplasm. Some microtubules of the spindle remains attached from one pole to the other pole are known as continuous spindle fibers, whereas some microtubules of the spindle remains attached to the chromosome and these are called as chromosomal spindle fibers. The chromosomes are the shortest and the thickest during this stage. RNA synthesis stops during metaphase and anaphase. In this figure, we can easily understood that chromosomes are arranged at the equatorial plate and spindle fibers are also formed, whereas the centrioles and spindle fibers we can identify easily. And it starts the anaphase. In anaphase, centromere replication takes place during these two sister chromatids, and due to that these two sister chromatids separate and travel to the respective poles. The separated sister chromatins are now called chromosomes. The daughter chromosomes towards that opposite poles are achieved by a contraction of chromosomal spindle fibers and the stretching of interchromosomal or interzonal fibers. During this, the chromosome appear as a rod-shaped or it is called as acrocentric, L-shaped or submetacentric or V-shaped metacentric. End of the anaphase is marked by the appearance of clusters of chromosome at each pole of the cell. Spindle fibers start shortening and pulling the chromosomes to the opposite poles. These sister chromatids separate at the centromere and move to the poles. Then the spindle fibers contract and the chromosomes are pulled to the opposite poles towards the centrosomes. Here in this image we can easily show that chromosomes moving to the poles and somewhat they reach the opposite poles. It is the final phase of mitosis. The chromosomes begin to uncoil their strands and become indistinct. The nucleolus starts reappears. The gel state of spindle reverts to the sole state and the spindles starts disappearing. The nuclear membrane reappears around the group of chromosomes and nucleolus, forming daughter nucleus. Chromosomes uncoil and becomes invisible nuclear envelope reappears, spindle fibers disappear, occurs simultaneously with cytokinesis, or single telophase takes place and after that cytokinesis takes place. Daughter cells have identical genetic information. In this image, we can see that chromosomes are uncoiled, nuclear envelope is reforming. The telophase, the cell actually divides. The chromosomes are at the poles of the spindle. The spindle disintegrates. The nuclear envelope reforms around the two sets of chromosomes. The cytoplasm starts to divide into two separate cells and the process of cytokinesis going to start. This is the cytokinesis. After nuclear division, that is a karyokinesis, division of cytoplasm, that is cytokinesis, occurs. In plants, cell plate is formed between the daughter nuclei. Endoplasmic reticulum and dictyosomes or Golgi complex move towards the equator and fuse to form the cell plate. Thus, two daughter cells are formed from the parent cell. Each daughter cell is with single nucleus having same number of chromosomes as that of the parent cell. Each daughter cell enlarges in size till it becomes comparable to the parent cell. In some plants, telophase and cytokinesis simultaneously takes place. The organelles other than the chromosomes get divided up into the two daughter cells. In plant cells, a new cell wall made of cellulose forms between the two new nuclei, about where the chromosomes lined up in the metaphase. Cell membranes form along the surface of this wall. When the new wall joins with the existing side walls, the two cells have become separate from each other. This is the outcome of mitosis, whereas two daughter cells, each containing identical genetic information, takes place. Daughter cells have the same number of chromosomes as that of the original parent cell. This is the overview of mitosis. There is after interphase in which chromatin material is totally uncoiled. In late prophase, the nuclear membrane and nucleolus disappears, and in metaphase, these centrioles to the opposite poles form spindle, and these chromosomes are lined up on the equatorial plate. In anaphase, these two sister chromatids separate from each other and the contraction of spindle fibers takes place, due to which the chromosome starts moving to the opposite poles. When they becomes reach to the opposite poles, immediately that starts uncoiled, and in between that cytokinesis also takes place, and thus there is end of mitosis takes place. This is the significance of mitosis. It leads very importance in the living organism, in which production of large number of identical cells for growth and development of organism takes place due to mitosis. Replacement of old, decaying and dead cells, bark formation takes place. Regeneration of damaged tissues and organs, that is a healing of wounds, takes place due to mitosis. Production of new organs, that is a new roots, new branches, takes place due to mitosis. Production of progenies identical to the parent plant, that is a vegetative propagation, is the very important role of mitosis. Responsible for qualitative and quantitative distribution of hereditary material to the daughter cells due to mitosis. Mitosis maintains constant chromosome number in a species. It also maintains the nuclear and cytoplasmic balance, which is called nucleoplasmic index in the cell. Now we see the video of that mitosis. Mitosis is a separation of nuclear chromosome into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, Anaphase, Telophase, and cytokinesis, in which cytoplasm divides into two. Before cell enters mitosis, it undergoes period of growth and rest. This period is called interphase. In interphase, the genetic material of cell increases. In prophase, the chromatin material becomes condensed and thick to form chromosome. Each chromosome has two sister chromatids joined together at centromere. The nucleolus disappears and nuclear membrane starts to break down. The centrosome, each with a pair of centrioles, move apart to the opposite pole, forming a spindle between them. During metaphase, microtubules of the spindle interact with chromosome. This causes the chromosomes move in the line of middle of the cell over the equatorial plate. Sister chromatids are held at the centromere, conforming the protein structure called kinetochore. Kinetochore holds the chromosome and attaches them to the spindle. During anaphase, the sister chromatids separate at the kinetochores and the daughter chromosomes along the divided centromere move towards the opposite poles as the microtubules shortens. The poles of the spindle also move apart and helps to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered as completed. During telophase, the daughter chromosomes arrive at the pole and chromatin starts to decoil. The nucleolus reform with nuclear membrane. This triggers the recognition of membrane with the two daughter nuclei. Cytokinesis is a partition of cytoplasm. In plants, new cell wall formation is triggered at the center of the cell by formation of cell plate. The cell plate grows and makes the existing wall into two daughter cells. Thank you very much for observing this video.